So I would suggest we start.
Mm-hmm.
Okay.
Mm-hmm.
So good morning, actually good evening, good afternoon to everyone. Welcome to the Phenomics webinar. Very much looking forward to the webinar by Jörg-Peter Schnitzler on the secret smell of plants. How can we use it for plant phenotyping? So I'm very much looking forward to the presentation, but let me very briefly first explain you some details about the Phenomics webinar series, what we do and what we aim to do. So the Phenomics webinar, it's organized mostly by EPPN emphasis IPPN with support of Nebraska Lincoln University and a platform in China. The basic idea is to provide an exchange platform for the community, mostly because of the COVID-19 related limitations of traveling and meeting in person, to meetings, webinar, to workshops, conferences. So we thought it would be useful to have an exchange via this kind of a web-based platform. We have two different formats for the webinars, single talks, thematic talks. And we depend actually on you, on basically everyone. The platform, the approach, it's open like at any conferences that you can submit your abstracts. When you go to the website, there is a submission platform. Please feel free to submit your abstract on any topics related to plant phenotyping, basically independent of your career level. It's from early career researchers to established scientists. Please feel free to submit your applications, submit your abstract, and we are happy to select you for the next talk. So the webinar series takes place every second Friday at 2 o'clock, with one exception. So we will go after this webinar, we go into the summer break. So the next webinar would be on 4th of September 2020. So don't miss the date. It will be an interesting talk. Very briefly, who are we? So why do we do this? So the webinar is organized, one of the organizers is IPPN. IPPN is a global network providing exchange platform for plant phenotyping centers and users from across the globe, with a mission to integrate the globally fragmented activities, enable, facilitate exchange of knowledge, information, expertise, address technology development, and provide advice to decision makers on state-of-the-art future opportunities and limits that are related to plant phenotyping. Then EPPN 2020, it's a EU-funded project with one of the key elements, which is providing transnational access. So there is currently the sixth and final call, which is open for any users to apply for access to one of the 31 facilities within this project. It's based on a rather simple application procedure, and once your application is approved, you get full cost coverage, including travel, and this includes also 20% of funds from non-European labs. So outside of Europe, you are open to apply. So far, the output was pretty good. We had applications from 34 countries, and 124 ongoing projects are basically in progress, and we expect at least 30, potentially 40 additional projects in a final call. And the output in publications, it's also pretty remarkable. So 70 publications resulted so far from the EPPN 2020 experience. Additionally, there is also the project or research infrastructure called Emphasis, and Emphasis aims at providing a long-term sustainable service provision with respect to phenotyping activities. So basically developing plant phenotyping infrastructure in Europe and providing access to multi-scale facilities and controlled environment field, as well as data and modeling approaches, then enable genotyping of performance in diverse environments. That would be the goals, and share knowledge and novel technologies that will be established within the Emphasis research infrastructure, which is an S3 project. The webinar, it's supported by AgriForm. So this is a platform which is in China. So ni hao to all colleagues in China. And it's also supported by the Nebraska Lincoln University, providing the technical support in broadcasting the Zoom webinar. And some very simple rules, which are pretty straightforward. So please put your questions in a chat. This is already in a chat. There is a request from Philip. So he's sitting in the background and reading your questions. So please put your questions into the chat. You find this chat button at the bottom of your screen. And at the end, potentially, if we have some time, we can also open a discussion, not only based on a chat, but also on if you raise your finger, you can also ask your question. The other point is we will have a break in the middle after 15-16 slides. So there will be time in between to ask some questions. So feel free to put your questions anytime during the presentation. One point, so talking about the presentation, so our speaker today, it's Jörg-Peter Schnitzler. So this is a short CV, so I tried to just highlight some of the few major achievements, but in the end, the text ended out in almost completely, bold. But anyway, so Jörg-Peter Schnitzler, Yogi, he studied biology in Tübingen in Germany, did his PhD in 1992. He went then as a postdoc to Munich, and where he's developed his interest in tree stress physiology and environmental simulation. Then he went to the Fraunhofer Institute for atmospheric research. And became interested in biosphere-atmosphere exchange with strong focus on volatile organic compounds. This is what he will also talk today about. Then he returned to Munich, and he's currently supervising one of the most advanced phytotron facilities and building, in addition, new phenotyping platforms for the study of plant genotype environment microbe interaction. And his lab investigates currently volatile, non-volatile metabolic networks in plant microbes under stress and the biological and ecological functions. So we are very much looking forward, Yogi, to your presentations, and as such, the screen is yours.
Yeah, Roland, thank you very much for the very nice introduction, and I'm very happy that I became invited to give a presentation in the IPPNet webinar. And for me, it's a new experience, not talking on real conferences, and so I'm really excited to learn a bit how these type of webinars and online lectures will work. I'm very happy to talk today about the secret smell of plants and how they can be used for plant phenotyping. And before in my presentation, I will start giving you an overview about what's the smell of plants, what are the compounds which are related to this smell, how they are synthesized, what's the diversity. And then we make a break, and then after the break, I will show you how this phenotyping can be done technically, what kind of infrastructure you need, and how you can perform experiments to get variations of smell of plants. So first of all, why is it looking? First of all, I have to introduce the smell itself. As you see here, so when we talk about volatile organic compounds, we are aware they are occurring in many different very different environments. We talk about the human well-being, we talk about smell and perfumes, we talk about medical applications in phytomedicine. So we are, as humans, very aware of all these type of molecules, and they are for our pleasure, they are for our food. So they are very important when we talk about our human well-being. And on the other hand, when we talk about atmospheric chemistry, they are strongly influencing the chemistry and the composition of the atmosphere. And in a very well-known example, it's the so-called blue haze above forest canopies. When you in some areas in US, you have the blue smoky mountains, you have these type of blue smoky mountains also in Australia. So depending on the solar angle, you can see a so-called blue haze above the canopy, and that's resulting from the oxidation of terpenes, which are released by vegetation. They get oxidized, and then they form small particles and aerosols, and then you get a change in light scattering, and that causes this type of blue haze. So that's a typical situation which you can have in an undisturbed area, and in that case, when you go to Beijing is a big mega city and with a lot of air pollution problems. And as you can see here, that's a typical afternoon in August. In summer, as you see here, it's really dusty. It's very diffuse, and it's very full of aerosol load. And that's also related to a certain extent to the degradation of volatile organic compounds, which are coming from biological or anthropogenic sources. But more interesting in terms of biology are all the functions these type of molecules have in the interaction between plants and microbes, and between insects. So these type of compounds are very important for the attraction or deterrence of insects. They are used as defense molecules against herbivores, against microbes. And many of these compounds are playing roles as infochemicals or signaling molecules, which are informing plants by plant-plant communication, or within a plant about stress situations which can appear below ground or above ground. In the plant itself, at least some of the compounds are discussed to have antioxidative properties, in particular the terpenes, isoprene, mono and sesquiterpenes. They are often linked to the antioxidative status of plants. And so taking together, these type of compounds are really important for human well-being, important for environment, and they are really essential for the beautiful interaction of plants with the environment. And that's what we will talk about today, how the variation will somehow help to understand plant communication and plant fitness with respect to environmental constraints. So when we want to make use of this type of compounds, we have to be aware that we have a very high diversity of chemical structures, but there are many environmental factors which can influence the release of these compounds. And on one hand, we talk about so-called constitutive compounds. So that means these are compounds which can be stored in a leaf or can be stored in tissues. And very typical storage structures are the trichome hairs on leaf surfaces, are the resin ducts in conifer needles or in hardwood of trees. So these are constitutive sources, and from there, the compounds can be released just by evaporation. On the other hand, we have the so-called de novo synthesis. So that means we can observe on the constitutive situations de novo synthesis, which is mostly relevant for isoprene or monoterpenes. And on the other hand, we have a release of emissions which are related to biotic interactions mostly. And in that case, we talk about a set of induced volatile compounds, and they are coming from stress. The stress can be either abiotic or it can also be biotic. And a typical abiotic stress can be heat, temperature, fluctuating light, drought, flooding. And on the other hand, a biotic stress, we talk about pathogens, herbivore, or pollinators. And in both ways, you get a set of compounds which are normally not present in the plant, but they are released after induction. And we talk about a huge amount of different terpenes. We talk about lipid oxidation products. We talk about alcohols, phenolic compounds. So we do not have only a very simple group of compounds. We talk about a very broad spectrum of volatiles which can be released, and very prominent examples which you can find in the atmosphere or released by plants are ethanol, acetaldehyde, acetic acid. So all three molecules are biochemically linked tightly to each other. We talk about terpenoids. In that case, we have isoprene, which is the most prominent volatile organic compound on Earth. We talk about the monoterpenes and the sesquiterpenes, which are a little bit more or less, let's say, semi-volatile. And on the other hand, we have compounds which are coming from the phenolic pathways. The most prominent example is the methyl salicylate. We will talk about later because it's also very important when we talk about a signaling molecule, and methyl salicylate is one of the most prominent signaling molecules in plant communication. We talk about nitrogen compounds, which are released also from phenolic precursors, and they are very, very common and very toxic against to herbivores. And the very classical group of volatiles, which all of us already know very well. So when you cut the green in your garden, you immediately can smell the fresh odor of the hay, and these are the green leaf volatiles. So when you cut your grass, then you immediately get the release of these that's the reason why they are called green leaf volatiles. So by cutting them, you destroy membranes, and by the destruction of membranes, you get a breakdown of unsaturated fatty acids, and then you get these type of volatile organic compounds, which are very fruity, which are very distinct smell. So all of us know it already. So we have a high diversity of these compounds, but that's not all, it's only one side. And when we go closer and look into a cell, we have to be aware that the diversity of the compounds originates from a very diverse biochemical origin. So that makes the situation even more complex when you want to understand the physiology and the biological functions. So for example, acetaldehyde, it's coming from the primary pathway. It's maybe it's a byproduct of ethanol breakdown, and in that case you get a release from the cytosol. We have in many cases we observe a release of methanol, and methanol is a very typical marker for cell wall growth, because in the cell wall, when the cell wall is growing and expanding, you have so-called pectin methyl esterases. And as a product of the linking of pectin, you always get a release. So methanol is a classical marker. When you see high methanol emission, you know the cells you are analyzing are growing very rapidly. And as mentioned here, we have the so-called green leaf volatiles, which are coming from the breakdown of unsaturated fatty acids from thylakoid and photosynthetic membranes. And then when we talk about terpenoids, we have two different pathways. On one hand, the so-called MEP pathway, which is localized in the chloroplast, and it's also present in some bacteria groups. And in that case, you get from photosynthetic precursors, you get a chain of reactions, and then they are leading up to a so-called C5 intermediate, and that's the dimethyl allyl diphosphate or isopentenyl diphosphate. And these are the unique precursors of all higher terpenoids. And one of these precursors, it's used by isoprene synthase, and then you get the release of isoprene. But you also can get a condensation of these C5 units to C10 units or even higher units. And on the level of C10, then you get by so-called monoterpene synthesis, you get the release of monoterpenes. In the case of sesquiterpenes, we have a second pathway which are leading to the same C5 intermediates. That's a called mevalonate pathway, and at the C15 level, the cells have the substrate to produce sesquiterpenes. So as you can see here, we have a huge diversity in the compounds. We have a huge diversity in the biochemistry and the metabolic origin of these compounds. And that all contributes to the high complex pattern of emissions you can observe under different environmental conditions. So the challenge will be how you can use this diversity to figure out, to analyze fingerprints, which are typical for a specific situation. It can be a stress situation, it can be a phenotypic situation. So that's a challenge, and that's what we are talking about today, how we can use volatile fingerprints. And somehow at the end, the final goal will be that we get a barcoding system which we can use to genotype plant traits by volatile organic compounds. So the idea of using these type of compounds is not only restricted to plants. And when you go to literature, so these volatile organic compounds are used in many different scientific areas, in life science, in microbial science, for using as marker for stress. And they are getting more and more important when you talk about plant diagnostics, so they can be used as shown here in a review, and that's what we are talking about today, that we can use it as a non-invasive diagnostic tool in plants. But they are also very important in studying lung diseases, in studying asthma or cancer. So a person who is getting sick changes the auto profile, and that auto profile can really be traced, and with a specific signature, you have a tool which allows you to really see whether a person is sick or healthy. And the same is also working for fungi. So we are working also on fungal chemical diversity, and you clearly see, and that when you go out in the forest, you see, and then you smell on the fruiting bodies of fungi, you really can smell the difference between the different fungal species. So you see, so the variation in composition or strength of these compounds are used in manifold ways for diagnostic purposes in human microbes and plants. And I, in particular, will focus when we talk about later on phenotyping of plant pathogen interactions, because volatiles are involved directly as a central component of all response to biotic interactions. They are involved really in direct defense as repellent or attractant. They are involved in indirect defense as they are able to cry for help, to attract, for example, predators of herbivores. They are very important messengers when a plant is infected, and then the neighbor can perceive signals from the infected plant, and then they can realize that something happens in the neighborhood. And they are very important also to prime plant defense responses. So without having optical signal, optical symptoms, these the volatiles can prime and bring a plant in an alert stage, and then the defense response can be much faster and then lead to resistance. And we also know that there's a high variation in genotypic emission pattern. And well, an example how important that can be in that case, it's a paper from us together with Corinna Flood, how these type of monoterpenes are very important when we talk about this type of priming and acquiring of systemic acquired immunity. So you see, they are very important when we talk about communication and interaction of plants with biotic stressors. And on the other hand, they are individual non-invasive markers for phenotyping, because they can be used for monitoring pathogenesis. They can be used for identifying chemotypes, monitoring priming effects, and also show how plants communicate with other biotic trophic levels. And then it's very important when we want to understand, for example, carbon fluxes. So since they are volatile, they can easily be used to trace C13 labeled intermediates. And as an example, I will show you a little bit from our previous work how this chemical diversity in plants, or in this example, for example, oaks, are important and triggering different behavior in chemical ecology. So when you go in Europe, we often have herbivore outbreaks. In that case, these are forests in northwestern Germany, and as you see here, we have a lot of oaks which are completely defoliated, and other oaks are still green. So there seems to be a difference, and we ask the question, why are some trees completely defoliated and other oak trees are completely green? And then we did a survey over many years, and then we realized, because the variation of defoliation is changing from year to year, but across several years, it became clear that we have several trees which are heavily defoliated, and we have trees which are partly defoliated. And so we thought about what's the difference in defoliation, and why the difference occurs in nature. And in that case, we are looking on a herbivore which is called Tortrix viridana, and it's a green leaf oak roller, and that's really a very strong pest in Europe. And looking closer on these plants and looking what's happening in the interaction with the larvae of these herbivore, you realize that depending if you have a sensitive or whether we have a tolerant oak, the release of volatiles is different. In that case, I show the trends in emission by monoterpenes, sesquiterpenes. DMNT is a breakdown product of terpenes, and then we have methyl salicylate. And as you see here, we add the caterpillar, and they start to feed on the plant, and then you see after a while there's a change in emission. We have a diurnal variation, but what you see here, the sensitive oaks emit differently to tolerant oaks. So the sensitive oaks are releasing more of the DMNT, while the tolerant oaks release more sesquiterpenes, and also the monoterpenes are also more released by the sensitive ones. So that means depending on the phenotypic behavior, the release of the compounds are different. And when in chemical ecology, it's often not that one compound is important, it's often the mixture which is important. And that's impressed here. When you look on the mixture of these release compounds, you see that the tolerant oaks release much more sesquiterpenes, while the sensitive ones release more monoterpenes. So there's a change in the pattern between both. And that's reflected on the biochemical level. You can analyze the genes or enzyme activities, and then you can clearly see that based on the emission pattern and based on the enzyme activities, it's very clear that you can separate these two group of phenotypes which are sensitive or tolerant. And then you can do testing. These are very classical tests in chemical ecology. You provide autoprofiles to larvae in so-called white tube assays, and then you can see which smell is preferred by the insects. And as you clearly see here, the sensitive oaks are much more attractive to the larvae compared to the tolerant. So that means you have a biochemical difference, you have a different emission, you have a different behavior, and the insect responds differently to the different autoprofiles. So that shows you the ecological relevance and importance of the release of these compounds. It's only one example, but I think it's a very, very impressive one. And how now these type of compounds can be used in different ways of agriculture or in forestry. And since they are influencing so many different parts in the environment, they can be used in smart agriculture, maybe by using as agents to prime, to bring in plant signaling in smart agriculture, or by tracing some compounds which are released as a pre-diagnostic marker. Very often they are used in so-called co-cultivation system. You have two different systems. Very prominent ones are the co-cultivation of potato and onions, and depending on the smell and the attractiveness, there's an exchange of signals between both plant types, and then you get overall an increase in fitness and health of both cultures. And in terms of bioeconomy, in terms of bioenergy, one idea can be to reduce the very high emission of isoprene, which is very prominent in all fast-growing bioenergy plants like poplars, willow, Arundus donax, and miscanthus. So that means when you produce a higher masses of biomass by plantations, you load the atmosphere with these type of molecules, and then you get problems in air chemistry. So there are several ways how you can use and apply these type of molecules in agriculture. And just to show you a little bit, and that's a very nice review from Matthias Erb, which was published last year. So there's always a signaling crosstalk, and that makes it so complex when you want to understand the interaction between organisms based on volatiles. So you can have VOCs volatiles from insects. Today we talk about volatiles from plants. We have the emission of compounds which are coming from plants. They can be constitutive or then can be stressed. And then you have also the release of compounds which are coming from bacteria or fungi. So that means we have a lot of molecules which are coming up in an ecological environment and a setting. And so that means the composite of all these molecules gives you an integrated volatile Q. And the challenge for a plant is to integrate the different information which is coming from different sources with a different mixture, with a different time constant. But plants are really able to integrate all these different signals, and that leads to integrated defense response. And a very prominent example is when we talk about plant-plant communication. So in that case, we always talk about a sender. So that means it's a plant which are releasing the compounds. And on the other side, we have a so-called receiver, and that's a plant which is perceiving the compounds. And there are many, many ways which can lead a sender to release the compounds. It can be that we have infested plants, and then the signal can come from a plant of the same species. It can come from another species. You can have information coming from infested plant of other species, but also, for example, a abiotically stressed neighbor can be perceived by the central plant. We have also, when we talk about competition between for light or some nutrients, you can also get a competition and communication via the volatiles. And as I showed you before, when you cut grass in your backyard, you see that you get the release of the green leaf volatiles, and these mechanically stressed compounds can also be perceived. So there are many ways how compounds can be received by a plant, and the origin can be highly diverse. But how we can use that, and that's important when you talk about plant phenotyping, how we can use that. And in that case, it's a very nice review. And in that case, the authors try to compare different techniques which are used in plant disease detection. On one hand, you have molecular techniques, you have spectroscopy or imaging techniques, and then we have the opportunity to use volatiles as diagnostic markers. And so depending on one hand on the cost, depending on the accuracy of the method, depending on the speed of detection, so you have to select which type of analytical tools you would apply. And so many of these parts are already very well established, and for using volatiles as markers in a similar way as doing it for example, a PCR or some spectroscopy, we still need to develop further technology. But I think it's very promising to do that. So we do not know actually about the accuracy of the method. And one really important part is the cost. So depending on the instrumentation, it can be very cheap or very expensive. But it really shows that we have a lot of potential when you are able to use high sophisticated, high-end instrumentation. And that's a big advantage when we talk about online analysis. It can be very, very fast, as I have shown you before with the oaks. So I think now we are almost in the middle, and I would be happy to get some questions for the first part. Do somebody has urgent questions for me about the introduction and the biology after VOCs? Please ask.
So are there any questions?
So I would have one.
Yeah, go ahead, Philip.
One question, especially now based on the last bits of information you displayed here, Yogi. Would you say an application of the profiling of VOCs for high throughput plant phenotyping would be in the near future? Or, for example, you say imaging techniques, they allow rapid detection, but of course you cannot bring them to the field. But let's stay first on the indoors. Would a high throughput system be possible in the future?
It's depending what you want to measure. So what we are doing, we are measuring with high-end mass spectrometry and in a very high resolution and sensitivity. But what you can do, you can already use so-called electronic noses, which are in principle relatively cheap, and they are not detecting individual compounds. They are detecting or integrating a order profile. So using this type of electronic noses, it's easily possible to separate treated and untreated, or infested and uninfested. And so that's a diagnostic tool. In that case, you can get a yes and no answer. And in many cases, for example, when we talk about smart agriculture, it often it's enough to say yes and no. We have an infestation in a stand, or no, we have no infestation. So there are cheaper sensors available which are not able to separate individual compounds, but they are able to separate the profile itself between different treatments, or treated and non-treated. And there are a lot of developments now ongoing in that direction, because having machine learning approaches as well, you're really able to, in principle, what you do is imaging analysis. You do not have an image of a picture, you have an image of a volatile signature, and then by using machine learning, that's an upcoming more cheap approach to at least get a yes and no answer depending on the question. But I think for phenotyping, that would be something which can be done in high throughput. But on the other hand, you do not know the compounds, and on the other hand, you have a problem with sensitivity. But yes, it's depending on the question. So they are cheap solutions and they are very expensive solutions.
So I guess the quantitative ones would be then rather on the expensive side, right?
Yes.
Okay, thanks you.
So Jörg, I had a question. If you could say something about, in this scientific context, about the relative benefits and costs of doing targeted versus untargeted, because you're talking about doing mostly mass spec. So are most of the studies, do we know the type of volatiles we're looking for? And so we're focusing more on targeted type metabolomic studies or targeted volatiles, or are we doing untargeted and kind of pluses and minuses?
I think the ideal situation would be when you want to use an untargeted mode. So the best would be to develop a system which is fast and cheap, and then try to verify that with a very sophisticated system showing you the composition of the compound. So that would be the best. When you want to use non-target cheap systems, which are electronic noses, which are able to detect profiles and separate profiles by machine learning or pattern recognition, then it would be ideal to really analyze what are the difference in the composition. So the best way is to combine both. No? Use the expensive ones, the sophisticated instrumentation, to verify what's in your cloud, what's in the profile, and then you can use it simple tools for the diagnostic purposes. No? So the trick is to combine both. And in that case, you only need the very expensive machinery only to verify what you have in your more cheaper system.
Okay, yeah, thank you.
There is a question on, did you try to measure VOCs from wheat diseases? And if yes, how and which diseases?
I will, in fact, I will later show phenotyping of barley. And we also did phenotyping of wheat, but unfortunately the data are not fully analyzed. So I would be happy to share all our wheat data, but yes, we already phenotyped wheat following a pest infestation, and they are really culture or cultivar differences. And I will show later how the differences looks like when we talk about barley, but in principle, it's comparable to wheat. Yes, so you can also trace differences in volatile emissions in crops after infestation. Yeah.
Okay, thanks a lot. I would suggest that we continue now, looking also at the time, and there will be, I'm pretty sure, quite a lot of questions at the end.
Yeah, I hope so. Okay, so now I will go in the second part, and And the second part is related how to analyze or how to develop these platforms. But before going that, I will just show you how diverse some of these volatile traits can be. And in that case, it's a survey which we did together with University in Vancouver, analyzing the variation, in that case of isoprene biosynthesis across the natural habitat of Populus trichocarpa. And so the Populus trichocarpa is growing in southern Alaska down to northern California. So it's a very long range of distribution with very different type of climates where this tree species is growing in the natural habitat. And then what we did, we went to the Common Garden in Vancouver, and then we analyzed many of these individuals. And as you can see here, these are the log-fold changes. And as you see here, you can use, it's in this case, it's only one trait, it's isoprene, but as you see here, really depending on the origin, you get a huge variation in this trait. So that shows you how it's very promising to use volatile markers when you want to look on a complete entire population. And that's an example of what we did with trees, looking on a huge population of trichocarpa genotypes. So just an example how diverse the volatiles can be. And now, before we talked about the biology and how they are important when you talk about abiotic and abiotic stress, and in the second half of the presentation, I will talk about the technological challenges, how to set up such a system, what you have to be aware, how that can be designed, and in particular, how afterwards you analyze all the data. And so Roland already mentioned, we are also part of German, European, and international phenotyping networks. And within the German plant phenotyping network, we developed a concept, and later on I show you we developed a phenotyping platform which enables that we analyze not in high throughput, but for volatile analysis, it's high throughput. In other terms, I think it's more medium throughput. How we are able to analyze and trace the emission of many plants in parallel. So we developed a concept where we have multiple cuvettes, and each cuvette is set up with the same design, and then they are put all together, and then you have to decide, and that's what we talked before in the round of question, what type of analyzers you will use. And in our case, we are interested in plant biotic interactions. So in that case, we thought about it's important to have an NO analyzer, because NO is a very typical stress signal when a plant is getting infected or infested by pathogens or herbivores. Then you always get a very strong release of NO, and then so with an NO signal you can really trace initial responses of plants toward the pathogen. And on the other hand, we decided we do not only want to have information about volatiles, we also thought about when we do these type of cuvette measurements, we automatically get all information about photosynthesis. So that means such a volatile organic phenotyping platform is not only a platform for VOCs, it's also a platform to phenotype photosynthesis. And even to go beyond classical only looking on that assimilation or evapotranspiration, we thought about now also including a stable isotope sensitive instrumentation, so that we can also distinguish between some CO2 which is maybe released during the night. But these are the add-ons. No? When we talk about volatiles, you have two options. On one hand, you can use GC-mass analysis. GC-mass analysis it's time consuming. You have often to trap the compounds on cartridges, and then you have to analyze online. Another opportunity is to use so-called online mass spectrometry. In that case, we talk about the so-called proton transfer reaction mass spectrometry. And that's a system which allows in online to trace volatiles down to a PPT level with a very high resolution. And in parallel, we decided to develop a fast GC system, which we are able to use then and trace these compounds by GC-mass as well. And doing a combination of all these analyzers, you get almost what at least in life science is called volatilome. So volatile means that's the release of compounds or the entire spectrum of compounds. We have nitrogen compounds, we have CO2 water, we have the green leaf volatiles. We can trace the different terpenes. We can trace the shikimate acid pathway derivatives like methyl salicylate, but we also can trace very short compounds. We can also analyze ethylene. So depending on the analytical tools, you're really able to trace many compounds, but you also have to be aware that's really challenging in terms of running these systems, and also challenging in terms of analyzing the data compared to simple systems like an electronic nose. But you have advantages, and as you see here, we have also disadvantages. And just to show you when you want to decide what type of method you would like to use for this for plant volatile phenotyping, you have to think about, we propose, or we started using online mass spectrometry. In that case, the pros are that we have a very high time resolution. We can really monitor with a very high also mass resolution everything what's coming out from an air sample. And that enables the annotation of some formula. We are extremely sensitive, so we have a limit of detection depending on the averaging and integration time, low to pp10, so parts per trillion. That's really very sensitive. Not as good as a nose of a dog, but already very sensitive. And we are able to detect excellently low molecular weight compounds. On the other hand, we have a limited accumulation time. So that means when you accumulate compounds on a cartridge, you can increase the sensitivity. And the big disadvantage of that technique is that since we are measuring masses, we are only measuring masses and have not a really clear identification of the compound. So that means we have to look for unique masses, and from these unique masses, we are able to derive some formulas or some annotation. So that's a limitation. And yeah, and the biggest limitation is that we are not able to separate mono and sesquiterpenes, because all isomers have the same molecular weight. So in that case, we only can say whether we get an increase in mono or sesquiterpenes, but we have no information about the spectrum of these compounds. So that's the disadvantage of that technique. And on the other hand, you can use gas chromatography, and in that case, you can accumulate the compounds on cartridges. So that means you increase sensitivity. And we already have very well established databases, NIST, Wiley, Kovac index, retention time indices. So we have a lot of tools which allow us to annotate all the compounds. The contrast in that case, we have a very relatively low time resolution, and the analysis, as you can imagine, you have to analyze chromatograms. It's more time consuming. And that's really depending on the column, and it's also depending on the absorber you use in the cartridges to trap the compounds. Depending on the chemical structure of these compounds, you are not able to trace all the compounds. So in principle, you have to trap the compounds on different absorbers, and then you get a more broader spectrum. So you are limited in the number of compounds you can detect. So the best way would be, in this case, and that's what we are doing, we combine both. So we use the advantage of the fast online monitoring and combine it with a high analytical resolution of GCMS. And combining that, you get really a volatilome from plants. Yeah, so developing cuvette systems from scratch it's really astonishing. It takes time. You need good engineers, a good workshop, but you are always faced by some unexpected problems. In that case, here since it's a closed system in the cuvette, we have irrigation. In that case, the water inlet was broken, and in a very short time, as you see here, we have a submerged. It's like an aquarium. We got a complete filled cuvette. So that's something which you not expect, and in that case, you can be very lucky that you do not destroy your detectors. So that's something which can happen when you develop new systems. But there are always, for all these type of problems which you have to face, it's a homogeneous airflow, gas tightness, gas turnover, no condensation, inertness of materials, high alert permeability of the cuvette, minimizing absorbance and so forth. So for all these type of problems, somehow there's more or less good solutions to really minimize artifacts and to really make sure that everything what you are measuring is really real and related to the best fitting and setup of the system. And that's how such a system looks like. What we did in Munich, we decided to always make blocks of 6. So we decided to make fully autonomous cuvette blocks of 6 cuvettes. So each block consists of 6 cuvettes, and each block has own flow controllers, have own clean inlet air, has clear separation of outlet line, and then you need multiplexing. And with the multiplexing, you can really always analyze individual cuvettes, because it's very impossible or almost impossible to buy too many of these mass specs. So that means in that case you have to move from one cuvette to the other one. And then you see we have all these GC tubes which allow us for GC sampling. So what we did, we constructed these blocks of 6. We programmed them, they are fully automatic, and then you can multiply these type of blocks. So what we did, we multiplied that by 4, and so finally in our final setup, we have 24 cuvettes, which are fully automatic running. We have volatile organic compounds analysis, we have a photosynthetic gas exchange, we get the isotope pattern, and we get the NO emission. So we get an entire profile, what's coming out of a plant under different types of stress. And that's a picture how it looks like. So we install these cuvettes in our climate chambers. So that means that we have a controlled environment. So that means we can really rely that all the different plants in the different cuvettes are treated in a similar way. And these are examples from our wheat experiment, which I will show you a little bit the data later on. But these are a closer look. And in that case, you have a glass cover, we have the plant. And in that case, that's a problem. In that case, with a barley or a crop plant, you're not really able completely to seal the soil from the above ground part. So that means in that case, you always have to run an empty pot with soil as a control. Otherwise, you are not correctly subtracting the background, which can come from microbes which are associated to soils. And so we developed a complete workflow how you can analyze the volatilome of plant phenotypes under highly controlled conditions. You have to grow them under very constant conditions. And since we are not able to cut the plant, so you need something which you use for normalization afterwards. In that case, we developed an imaging station. So before putting plants into the cuvette system, we have an imaging station where we make 3D images from the plant. And then based on these 3D images, we get the leaf area, and then we lead later on to normalize the emission rates. We have to put the plants in the installation. We measure, allow that the system is stable, and then we can follow up for at the moment we're always running for 6 to 7 days. And then you get a lot of data. You have diurnal data, you have data over 1 week, you have treatment data, you have data from background. So that means you need a computation pipeline which allows you really to analyze all the compounds in a reasonable amount of time. To show you the first experiment, what we did in this system, we did in this system not wheat. We decided to make an initial experiment with a plant which is called tansy, Tanacetum vulgare. Tanacetum vulgare is an aroma plant which is very common in Europe and in Eurasia. And it's a plant with a high chemical diversity in terpenoids. And on the left side, you see we selected 5 plants. And what you see here, these are the terpenoid chemotypes. So that means each plant smells different. Really, each plant has an individual odor profile, just to see how extreme the emission can be. And then we put them in a cuvette system. And on one hand, we started. And then after 1 day, we added aphids. And the aphids are sucking on the floor, but in that case are a low invasive herbivore. And then a couple of days later, we added in some of the cuvettes, we added a real leaf caterpillar feeding the plants. And And that's how it looks like here. That's a plant in nature, and these are Spodoptera littoralis. It's a moth, and as you see here, they are very hungry. So we only put in two larvae. So that's a result of feeding for only 2 larvae, and they are really just almost destroying the plant. But that's a good example what can happen afterwards with emission, and that's shown here in this example. And these are the online traces from one plant. It's only one plant, just to show you how this online technology can be used to trace, in that case, insect behavior. And I here show you the release of the green leaf volatile. So that means when a leaf is fed, you immediately get a breakdown of lipids in the chloroplast, and then you get an immediate release of the compounds. And as you see here, when we add the larvae to the plant, they immediately start eating, and each bite leads to a release of green leaf volatiles. And then you see there are periods where they are non-active. So that's a way how you can use, in this case, to trace insect behavior. It's something which is completely different what we are talking about, but you can use, in this case, the green leaf volatiles to really see the activity of the individual caterpillars. And then you can imagine that this behavior maybe is different from plant genotype or phenotype to the next one. When you want to compare the different phenotypes, we did an overall analysis with 6 replicates, and always these are the traces which we get from online mass spectrometry. And as you see here again, adding of aphids. In that case, we added 100 aphids. You get almost no change in the emission. But what you get a very strong response. In that case, when you add the caterpillar larvae, you get a huge emission of monoterpenes. You get emissions here, also an oxygenated monoterpene, here another one. And so you get a bunch of compounds which are released by the treatment. And to see here, we see also difference whether the plants are previously primed by aphids or not. It shows very nicely the capacity of online mass spectrometry, but on the other hand, it also shows the limitation, because for many cases, we are not able to separate the compounds. And that's, for example, here, when we talk about the monoterpenes or sesquiterpenes. And for that, you need different tools. In that case, you need GCMS. And when you combine GCMS analysis of the individual terpenes which are in the trichoma, which are stored in trichoma, you see the different phenotypes. When you use online mass spectrometry, even with a limited resolution of monoterpenes, you also can see the different phenotypes. You can also see that they are different in response to the different treatments. And then when you add again GCMS on the volatile profiles, you again can see the 5 different genotypes, and you can also see the treatment. So I think this example really nicely shows you the capacity, but also the workload, what you get when you try to use this type of phenotyping for plant biotic interactions. But I think it's very promising because you can really go deep and dig in maybe later on biological functions. And we already had a question about crops. And that was the second experiment which we did in our facility. In that case, we used in our German plant phenotyping network, we had a collection of reference barley cultivars, and we used the reference barley cultivars from Jülich and Gattersleben and used them as target for powdery mildew infestation. And as you see here, after a couple of days, here you see the symptoms of powdery mildew on the lower leaves of the barley plant. And following the emission of volatiles in these 10 barley plants, that's shown here. In this case, I only selected 2 compounds, monoterpenes and methanetiol. And here you have these 10 different cultivars. And as you see here, for the monoterpenes, we have varieties which are almost not responding. We have varieties which respond very, very fast, very early. We have varieties which respond very late. So as you can see here, clearly we have a diurnal variation, but we have also the variation between cultivars. And that makes it so interesting, so that we have cultivars which are always not responding. Maybe they are resistant, as I will show you later, or some are responding very rapidly. And it's even better to see when you look on methane ethyl. It's a very simple molecule, and in some cases it's almost absent. But in some cultivars, as you see here, they are released in very reasonable amounts. But again, we have always to keep in time. We have to be aware that time is important. So you cannot only analyze one day. So in that case, when you want to do plant VOC phenotyping, you always have to look on the emission profiles of several days, and that makes it a little bit difficult when you want to do fast, high throughput phenotyping, because you have to know. So, for example, having a quick sensor and you do plant phenotyping at day 6 or 7, no? You get a totally different profile when you do the same at day 3. So that means before you do quick high throughput phenotyping on VOCs, you have to know the time frame and the response behavior of the plants. Otherwise, you maybe are at the total wrong time point. And in this case, just to almost finish my presentation, I show you here the variation of different groups of compounds in the barley cultivars, and these are so-called radar plots. And I mark the plants which are more or less resistant to pathogens. So here the red ones are completely resistant. We have moderate susceptible, we have late resistance, penetration resistance. So we have different types of resistance or susceptibility. And then you can try to link the phenotypic behavior to the emission profiles. And that you can clearly see here. So a plant which is maybe totally resistant, then you do not get a release at all. So that means it's not only necessary to know the emission profile, you also have to phenotype the overall pathogenesis and the plant behavior, because only then you can really relate the emission profiles you get from VOC screening and the pathogenesis. Good. Ending up, what are the challenges? So I think when you want to, or I try to convince you that it makes sense to use volatiles for plant phenotyping, but there are a lot of challenges which you face when you want to do that. On one hand, you have to think about the cuvette design, because most of the systems you have to develop in your own. So you have to be aware how to do it and when you do it. And so that's one, the hardware is one point which you have to consider how to develop. But then the next step is how to make the software for running the system automatically, and in particular to develop tools for data analysis. And what we are now doing, we are applying machine learning approaches to make it more simple and more fast. But that's something which you have to be aware in mind when you do omics. And in that case, it's volatile omics. We talk about huge data sets, and in that case, it's very important that you develop analytical tools which help you to analyze the data afterwards. And that's the most challenging part, at least for us at the moment. We have now the system, but the most challenging part is how you speed up data analysis. You have to know all the physical properties, the air, everything must be correct and somehow controlled when you want to get comparable data. And yeah, and that's a question whether in some ways it makes sense to use online mass spectrometry. In some cases, it's maybe better to use GCMS as an offline tool, or use electronic noses or other cheap sensors for high throughput. And when you always have to be aware, when you run sophisticated mass specs, you have a high maintenance of the system. That's really something which takes a lot of time. So these are all challenges. But finally, I think I could show you that when it's working, you get a very nice, exciting tool, which you can also apply to crops which are very low emitting compared to oak trees, or when we compare it to tansy. And yeah, if somebody is interested in looking how the system is working and maybe interested in using it, you can go to our website where you can find the link and information. Finally, yeah, it's a huge work load to develop such a system. In that case, I have to acknowledge a lot of people. And on one hand, the system was initially set up by Bijou. She was a postdoc for the beginning of the project, and the next postdoc was Van der Jood. So they both together set up the mass spectrometry. Barbara Winkler, she's a highly experienced person in photosynthesis, so she was very crucial to set up the cuvette system. And then you need a lot of stuff coming from my lab, coming from other institutes, and these are the cooperation partners for the different projects. And yeah, finally you need the money. In that case, we're very happy that you got the money for all the instrumentation by the German Plant Phenotyping Network, and the money for DPPN was coming from our Ministry for Education and Research. And yeah, German Science Foundation is always needed when you want to get additional money. And yeah, I am happy to take your questions.
Thanks a lot, Yogi, for the great presentation. So there are some questions. In case of barley, which respond faster, the resistant or the susceptible?
The susceptible. So I think in the case of barley, so you can, there are two ways how you can get the response. So one can be that the plant is primed, and then you get a release in terms of an alert emission. And the other one, that it's really a defense-related response. So that means when necrotic area starts to develop, when the chloroplasts crash and the cells break down, then you get a release of compounds. So in that case, it's a response, and not so you can distinguish between a clear response, which is really related to the destruction of tissue, and on the other hand, you can observe an emission which is related to the genotype. So that means it's also depending on the compound. So for example, when you observe green leaf volatiles, they are very strongly indicators for destruction of tissue, because they are released from unsaturated fatty acids when cell membranes break down. So that's a clear indication, and there you can really make correlation between increase in green leaf volatile emission and destruction of leaf tissue. So they are strict correlation. So these compounds you can use maybe to monitor the development of necrotic area. When we talk about mono and cis-quiterpenes, in that case, they are a response to a trigger. In that case, they can be a cry for help, that in case of an herbivory, for example, the plant is crying for parasitic wasps, which are feeding on the herbivore, or they are an indication of early stress response. So when we treat a plant without any symptom, you already get a release of compounds which are important for plant-plant communication. And in that case, you do not get symptoms. So that's something which you have to take in account when you analyze the data, that some of the compounds are related really to a destruction of the tissue, and some of the compounds which are released maybe are showing a constitutive difference, and other ones are more related to signaling. And that makes it complex, but on the other hand, it opens the opportunities to distinguish. But in that case, you need really information about the pathogenesis, if the cultivar is susceptible or resistant.
Mm-hmm. There is a question on eNOS. Could eNOS provide us what information? What kind of information could an eNOS provide us by integrating with basically the techniques you use for GC and PTF for detecting volatile organic compounds?
So eNOS are the principle for eNOS. You have a surface. It's more or less a semi-lipophilic surface, and volatile or compounds can bind on the surface, and then the surface is under current, and then you get a change in voltage. So what you do, with an eNOS, molecules bind, and then the current which is in the membrane or in the surface is changing. And what you do, you measure changes in current. And depending, and many compounds can bind on the surfaces. No? So that means you get, what you only get, you get a change in the current. And that allows at least to compare a treatment and a non-treatment. For example, you have a healthy plant. And a healthy plant is normally not emitting mono, or many plants are not emitting mono or cis-terpenes. And then you treat the plant, and then you get a release of monoterpenes, and then you use an eNOS. In one case, you get a very weak signal. In the other one, you get a strong signal. So that means the eNOS allows you a yes or no answer. No? It's not telling you about the composition, but it tells you, yes, I have an infestation.
Could you calibrate your eNOS in a way that you, when you know your specific crops, you know when those plants are infected, and use it in the end also for precision agriculture, and would it work?
So there are a lot of developments in electronic noses. So the biggest problem with electronic noses, they are not very sensitive. So the best electronic noses go down to 1 ppm or a little bit lower. And as I mentioned before, online mass spectrometry goes down to 1 ppt, parts per trillion. And when with GCMS, you come down with 1 ppf. And in the case of electronic noses, we are 10,000-fold less sensitive. So that means you need very strong emissions. That's a problem. But on the other hand, they are very simple advices. And I have seen electronic nose developments. In that case, you can make a clip. So many people try to develop clips. You have a very small, smart electronic nose. You can put it as a clip on a leaf surface. And then you have an electronic signal which is sent out in a cloud, and then you can put these type of electronic noses on a leaf surface, and then you read in the cloud always changes in the current, and then you can apply multiple of these sensors directly on leaf surface, for example, in a crop stand, and then you can analyze changes in the electronic nose signals on a spatial resolution and on a time resolution. And in that case, when you really put it on directly on a leaf surface, you also increase a little bit the sensitivity because you are directly at the site of emission. And in that case, there are really promising examples that you can use this type of small, in principle, we talk about wearables. So there are several ways how you can try to develop electronic noses as wearables. And then it might be very, very helpful in agriculture for sensing pest infestations. So there are many ways how you can use it. And in that case, again, you only ask for a yes or no question. Calibration, I think it's not possible.
Okay, yeah, thank you. So there is a interesting question about the difference between young and older plants with respect to the VOC profile. How big are the differences? Are there any differences?
I think you have not to talk about young and old plants. You have to talk about young and old leaves. So, as mentioned in the introduction, when cell walls are expanding and growing, you always get a release of methanol because of the elongation of cell walls. So that means young developing tissues release a lot of methanol. They also release a little bit more of ethanol. And so you have a developmental dependent change in the emission profile. So young leaves have a different pattern compared to old ones. And that makes, for example, for isoprene emitters, so very young leaves of willow or poplar or birch, they release in the very young tissue, they release monoterpenes. And when the leaves get fully mature, the emission change from monoterpenes to isoprene. So in that case, the chloroplasts in young developing tissue produces monoterpenes, and when they get fully mature, they switch from mono to isoprene. No? And that's just something which is related to the carbon flux and the maturation of the chloroplast. So that means it's really not easy to say a young plant emits different to an old plant. It's really depending on the leaf tissue.
Mm. How do you deal then with whole plant measurements, because you get this mixture of young and old tissue, so you have a matrix of signals?
Mm-hmm. Yeah, that's a problem. So there are two ways how you can do that. You can go to use only a leaf part, no? Or you will enclose a complete plant. And we decided to look on the overall integration of the emission, because the emission is not only depending on leaf positioning and leaf aging, it's also depending on leaf temperature and leaf perception or light perception. So that means within a canopy, go back to the barley plant, we have different leaf angles, we have different temperatures, we have different light perception. So that means in principle, light and temperature trigger photosynthesis, and many of the compounds are released independent of photosynthesis. So that means when you go to see such a barley leaf, which is hanging down from the plant, each centimeter you have a different photosynthetic activity. So that means you can also have different emission rates depending on age and also on light and temperature perception. So that means it's very difficult to select which part of the leaf you are trying to analyze. And in that case, we decided we have to go to the larger scale to look at the plant itself. It's an integration, but on the other hand, it's the own way to get rid of the huge variation when you go to specific leaves or leaf parts, and then you have the influences of canopy shading and canopy light and temperature differences.
Mm-hmm. Yeah, thanks. There is a question, actually two questions that I could probably combine into one. Yeah, so there is a question about drought stress. So is there a good case study about drought stress? And the other question would be partly maybe also related. Is there an effect of VOCs and soil nutrition? It's not completely related, but still you get this drought stress, soil nutrition. What are the effects of those on VOCs? Yeah.
So when we talk about the emission, in most part of the talk, we talked about an increase in emission. No? But when a plant maybe faces drought stress, we can also talk about a reduction in emission. So it's not only that a change in emission must occur that the emission gets higher. It can change, but it also can get lower. So that means when you apply drought stress, many of the compounds or the emissions go down. When you apply drought or water limitation, the plants stop growing. So that means very rapidly the emission of methanol or ethanol or acetaldehyde goes down. That's a response to stop of growth. No? And on the other hand, you get an increase of leaf temperature because you have when you have low evapotranspiration, the intrinsic leaf temperature is higher. So that means emissions which are depending on biosynthetic activity, and activity depends on temperature. So that means you get an increase in temperature, and that means you get an increase in release. So that's an effect which is related to temperature, for example. And then when you apply very severe drought, and then you at a certain point, the chloroplastic membranes also start to degrade, and then you get also the green leaf volatiles. So drought can cause a reduction in emission, particularly of oxygenated compounds. For compounds which are depending on photosynthetic activity and temperature during biosynthesis, you often get an increase. And when the drought is highly severe, and then you get the destruction of membranes, you get a lot of compounds which are coming from degradation of cellular structures. Talking about nitrogen or fertilization. So we also talk about nitrogen-containing compounds. So that means, and yes, when depending on the nitrogen status, you get changes also in the emission rate. But what we observed in that case, we compared mycorrhizal plants and non-mycorrhizal plants. And the mycorrhizal plants has a better nitrogen and phosphorus nutrition compared to a non-mycorrhizal plant. In that case, when you stress these plants, you get a total different emission profile, depending on the presence or availability of phosphorus or nitrogen. So yes, nutrition also influence the emission rate and pattern.
Mm-hmm. Yeah. So maybe a question on the aphids, on the experiments with the aphids. Mm-hmm. So when you expose your plants to the aphids, so there is an effect probably of mechanical damage and probably a specific of the linked potentially to the spit or the compounds the aphid is producing. Can you differentiate between those? Or
so the aphids are in principle more minimal invasive herbivores. No, they are just trying to suck from the phloem. So the mechanical destruction is very, very small and very weak. But on the other hand, they inject some elicitors into the phloem, and these elicitors can trigger a plant's response. So that means you can get a change in the emission profile of a plant just by the signaling compounds which are released by the aphids into the phloem. So and it's also not clear whether an aphid smells itself. No? So that means it's in our cases, the plants were relatively big compared to 100 aphids. So 100 aphids sounds a lot, but when you look on the biomass, it's only a very, very tiny part of the entire biomass. In that case, we were not able to separate. No? But when you in that case, it would make sense to go to a small part of a leaf, or just a leaf petiole. In that case, it's maybe easier to trace emissions from the aphids or locally triggered volatiles from the plant. That's not possible on the big level of an entire plant. In that case, you dilute most of the signals. And that's different to the herbivores. When you see, when the larvae are really eating, then you get a huge emission.
Mm-hmm. And maybe, looking also at the time, maybe the last question. So there is a question, so once you identify a specific volatile organic compound from plants, so you obviously need to use mass spectroscopy. And once you identify a specific VOC related to disease, how would you use it for an early detection system? So basically when you know when there is a very specific compound, can you use it for early, it could be...
So a good example is, for example, maize, corn. Corn, when corn is infested by insects or pathogens, a very typical compound which is released under that biotic stress is caryophyllene. Beta-caryophyllene is a very, very prominent sesquiterpene which is released by most of the different plant species under biotic stress. And so they are a concept that you use beta-caryophyllene as diagnostic marker for early infestation by pathogens, in particular in corn. And that's a specific signature, so that you can easily or relatively easily detect by the electronic gnosis. So caryophyllene is a very classical marker when you want to see early stages of infections. Other early markers which are very prominent are ocimene and myrcene. So ocimene and myrcene are open non-cyclic monoterpenes, and they are typically released at the very early beginning of biotic stress. So caryophyllene is a very excellent marker for stress, and also ocimene and myrcene. They are both together are also very good markers at the very early beginning of biotic stress in most of the plants.
Mm-hmm. Okay, so Yogi, looking at the time, thanks a lot for the time. Really appreciated your talk.
So I can offer, if somebody has further questions, send me an email.
Yes, please feel free, send any questions you would like to, and Yogi is welcome to respond to any of those questions. The facilities maybe at some point also be part of the phenotyping networks initiatives within Europe, and potentially available for access to do the experiments, interesting experiments in there. And finally, maybe one last announcement just to remind you that we are going now into the summer break and we'll restart the webinar series in September. And the next talk will be by Roland Schaffleitner, phenotyping at the World of Vegetable Center. So he will talk about vegetables, and you are very welcome to join the talk. And by that, Yogi, once again, thanks for the presentation. Really appreciate it. And also enjoy the summer now. And as such, thanks a lot for the participation. Thanks a lot for many questions. And we are looking to see you back after the summer break. Thanks a lot.
Yeah. Bye-bye to all.
Thank you, Jörg.
Bye-bye.
Thank you. Excellent.